Firstly, thank you to the organizers for the opportunity to speak today. So I'll be discussing my work on IgE-Fated B-cell memory and how we can perhaps reprogram these cells so that they no longer take this IgE fate. So as a brief background into IgE memory, we know that in IgE-mediated allergic disease, IgE will bind to high affinity receptors on mast cells and basophils, and this can persist for weeks to months. Circulating IgE, on the other hand, has a very short half-life of less than 3 days in humans. And then the cells that secrete this IgE are actually short-lived relative to something like an IgG plasma cell that would that would arise following a vaccination. Those cells can be quite long-lived. So given that this kind of entire humoral arm of IgE immunity is relatively short-lived, we focus on studying the memory compartment or the cellular compartment, where both memory T cells and memory B cells are important in this process. So my work has specifically focused on how memory B cells ultimately give rise to this transient IgE arm that we see in IgE-mediated allergic diseases. So very recently, we and others have shown that IgE memory B cells are extremely rare or absent in allergic individuals. And instead, actually, what is maintaining that IgE pool are long-lived IgG1 memory B cells. So there's this requirement of a non-IgE memory B cell to replenish circulating IgE antibodies. As well, we recently found that a subset of memory B cells with a distinct type 2 polarized signature maintain this transient IgE response, and that their development is dependent on IL-4 signaling. And ultimately, when these cells are re-exposed to the allergen, they will produce this transient IgE B cell pool, which rapidly differentiate into antibody-secreting cells and will secrete IgE that ultimately can elicit allergic reactivity. So these IgG1 memory B cells have this type 2 signature, as I mentioned, which is denoted by high expression of the low affinity IgE receptor CD23, as well as high expression of the IL-4 receptor, which expression at that level is normally only seen on naive B cells. So what we were interested in studying is whether or not this type 2 polarized IgG1 memory B cell pool can be redirected away from this IgE fate. So to do this, the experimental approach that we used was a mouse model in which mice undergo tape stripping. So they're epicutaneously sensitized to this model allergen ovalbumin for 2 weeks and then are rested. And we know that this sensitization protocol elicits an IgG1 memory response as well as circulating IgG1, but the mice are not yet clinically reactive to the allergen, nor do they have any detectable IgE in their system. So this allows us to have this temporal control over the IgE response. So then we can provide non-sensitizing re-exposures, which drive this IgG1 memory B cell response into an IgE state. So now the mice will have detectable circulating levels of IgE, and they'll undergo IgE-mediated clinical reactivity upon allergen challenge. And in other work, we've shown that if you block IL-4 receptor signaling during these non-sensitizing subcutaneous re-exposures of ova alone, that you prevent the IgE response. But what we don't yet know is, well, what's happening to those IgG1 memory B cells that were established upon sensitization? Is it just that we're blocking the IgE response, or are we causing some fundamental change in the allergen-specific memory B cell compartment that was established upon sensitization? And so to study this, we assessed how IL-4 signaling blockade during the recall response impacted the transcriptomic phenotype of naive and allergen-specific memory B cells. We also looked at their BCR repertoire through sequencing, and we employed allergen tetramers to do most of this work, looking specifically within the allergen-specific compartment rather than the bulk B cell compartment. And in the interest of time today, I'll just briefly touch on these latter two. So what we observed with regards to their BCR repertoire and the allergen-specific response, both at the cellular and humoral levels. And throughout this presentation, the abbreviations IC refer to mice that received the isotype control, and A4RA is mice that were treated with anti-alpha receptor alpha during those non-sensitizing reexposures. So first we looked at the effect in vivo of IL-4 receptor alpha blockade on antigen-specific B cells. So using this fluorochrome conjugated tetramer approach, we're able to identify this nice population of antigen-specific cells. And we could see that the frequency or the overall number of these cells went down in anti-alpha receptor alpha treated mice. However, they did persist. So next we looked at whether or not their entry into germinal centers was at all impacted. And what we could see, so we did this by looking at CD38 and GL7 expression. So on the top left quadrant, you would have cells that are conventionally in a resting state. This bottom right quadrant are cells that are in a germinal center, and then this top right would be cells that are transiting either to or from the germinal center. So we followed this germinal center response out to 28 days post-reexposure. And at an early time point, so just 3 days post-reexposure, there is a substantial blunting of this germinal center activity with anti-alpha receptor alpha treatment. However, this response is no different by the time that day 7 rolls around. So we were able to observe that germinal center activity can still happen in the absence of IL-4 receptor signaling, although the kinetics of it just seem to be a little bit delayed. So then we looked at whether the memory B cells maintain this type 2 polarized signature that I mentioned in the introduction. And again, that's going to be this upper right quadrant with high expression of CD23 and IL-4 receptor alpha. And excitingly, we could actually see that by treating the mice with anti-IL-4 receptor alpha during those subcutaneous exposures, this pathogenic phenotype is quite blunted or significantly reduced. So given that we could still observe germinal centers in these mice, but the type 2 signature is decreased, we were left wondering what the product of this recall response is. So it's obvious that these memory cells are being reactivated. We know that they're not producing IgE. So is it that they're just continuing to produce IgG1, or is something else happening? So then we asked the question of whether or not there's an isotype bias or skewing with anti-alpha receptor alpha blockade. And we looked at the other isotypes that could be produced, and quite strikingly, there was this clear emergence of an IgG2c population with anti-alpha receptor alpha blockade. So these treated mice had a lower percentage of their class switch over specific memory B cells being IgG1 positive, and then this emergence of an IgG2c population. And this population did appear to also be long-lived, as we could find it even 15 weeks post-re-exposure in the mice. So of course, IgG2c in mice is associated with a type 1 immune response. So we were wondering if the development of these cells required interferon gamma. So to do so, we also administered co-blockade of secreted interferon gamma, and we could see that ultimately the emergence of this IgG2c cluster population was dependent upon interferon gamma signaling, leading us to conclude that this IL-4 receptor alpha blockade could induce the formation of IgG2c memory B cells in an interferon gamma dependent manner. So this shift to an IgG2c response was observed not only at the cellular level, but also within the circulating immunoglobulins So by ELISA, we looked at, again, OVA IgG1, and we know that the titers of OVA-specific IgG1 will never go back to baseline in an allergic mouse because those plasma cells can be quite long-lived. On the other hand, we have already shown that IL-4 receptor alpha blockade will prevent the emergence of IgE in a recall response, and so that was consistent here. Whereas in the isotype control treated mice, you get production of IgE, but it's actually quite transient. So those levels will drop off relatively quickly. And then looking at the isotypes that are upstream to IgE, there was no difference in the IgG2 B response. And again, we only see that 2C response emerge with anti-IL-4 receptor alpha treatment. But you'll note that the titers of IgG2c, unlike IgG1, actually appeared to be more similar to IgE in that they dropped off relatively quickly. However, you'll recall that we could see OVA-specific IgG2c memory B cells that were long-lived, and they persisted for out to 15 weeks post-re-exposure. So what we next asked is, given that we know that there's a long-lived cellular arm, can those long-lived memory B cells ultimately give rise or replenish this transient humoral IgG2c response? And to do this, we gave mice tertiary antigen re-exposures at 9 weeks, and this time without re-administration of anti-IL-4 receptor alpha. So just to clarify, the mice were sensitized. So they have a pathogenic IgG1 memory B cell compartment. We then treat with anti-IL-4 receptor alpha and administer non-sensitizing re-exposures. And then we waited 9 weeks for that blocking antibody to clear, and then gave additional re-exposures of OVA alone, this time in the absence of anti-IL-4 receptor alpha blockade. And strikingly, we didn't see a reemergence of IgE in those treated mice, suggesting that the cessation of the treatment didn't impact their redirected fate away from IgE. And instead, we actually could see the emergence, or the reemergence, I should say, of IgG2c as well as IgG2 B. Overall suggesting that this altered trajectory of allergen-specific cells was stably maintained even following cessation of the treatment. So next we asked whether the origins of these IgG2c B cells were de novo, so arising from naive B cells, or were we actually reprogramming those pathogenic IgG1 cells that we know are established upon sensitization? And so to do so, we bulk sorted out OVA-specific class switch, the OVA-specific B cell compartments, then performed PCR sequencing with the repertoire platform and got some high-level pre-processed data, which we piped into the Immcantation framework, first using ChangeO to cluster sequences into clones, and then finding a consensus germline sequence per clonal family, and then moving this over into Dowser to build those clonal lineage trees. So firstly, what we could see is with looking at the frequency of class switch isotypes, there was a higher proportion of IgG2c sequence or IgG2 sequences in anti-alpha receptor alpha treated mice, which is consistent with our flow cytometry and serum immunoglobulin data. Then next we looked at the mutations of these cells, and actually quite interestingly, we could see that IgG1 and IgG2 OVA-specific cells were more highly mutated in anti-alpha receptor alpha treated mice. And this could go hand in hand with some recent work with from Marion Pepper's group, which showed that IL-4 actually downregulates BCL6 to promote memory B cell selection. So perhaps in the absence of IL-4 signaling here, we're enabling these cells to undergo further rounds of somatic hypermutation, ultimately permitting them to be more highly mutated. And lastly, what we were most excited to look at with these sequencing data is whether or not we could observe expanded IgG2 clones having or sharing clonal ancestry with upstream isotypes. And in fact, we could see that clonally expanded IgG2 shared clonal lineage with all upstream isotypes. So naive cells expressing IgM and IgD, as well as IgG3 and even IgG1. Overall illustrating that the class switch origins of IgG2 clones are comprised of multiple isotypes, and perhaps indicating that in our in vivo experiments, we're not only getting the production of IgG2 from de novo naive B cells, but we could also be shifting those pathogenic cells that were established upon sensitization and redirecting them to an IgG2 fate. And of course, this was all in the context of anti-alpha receptor alpha. So lastly, we asked whether or not this can also be achieved using a type 1 adjuvant at the time of antigen exposure. So here we used an adoptive transfer model in which the donors were all immunized with alum, which is a potent type 2 adjuvant. And we transferred their over-enriched class switch memory B cells. And so you'll note here that almost all of the cells that would have been present in that transfer were IgG1 expressing. These were transferred into B cell deficient mice. And then these mice either received re-exposures with ovin cholera toxin, which is another type 2 adjuvant, or ovin CpG, which is a type 1 adjuvant. And we could see that with great exposure in the context of a type 1 adjuvant, we could get this emergence of an IgG2c memory B cell population. And then within the serum, there's no production of ova IgE when re-exposing with that type 1 adjuvant, but you do have the emergence of 2B and to a lesser extent 2C. So leading us to conclude that not only can we achieve this reprogramming with anti-alpha receptor alpha, but it can also be achieved using a type 1 adjuvant to redirect the fate of these type 2 polarized memory B cells. So in conclusion, we could see that in the absence or in the blockade of IL-4 receptor signaling during this recall response, we're able to significantly diminish this pathogenic type 2 signature denoted by CD23 and IL-4 receptor expression on IgG1 memory B cells. And we can also push these cells, as well as naive B cells, to undergo non-Ig class switch recombination. So in this case, we observed that their redirected fate was towards IgG2, and that the IgG2c cells that emerged in these mice were long-lived, and ultimately gave rise to transient IgG2c serum responses. And I didn't show it here, but protected mice from undergoing IgE-mediated clinical reactivity. So to wrap up, I'll just thank my co-author on this work, Elisa Phelps, all the other co-authors that help with the work, our collaborators at ALK, our blood donors, and our funding. And currently it's out as a preprint and is in review if you're interested in looking at it. Thank you.
Thank you very much, Kelly, for your presentation. There is a question on the chat, and in the interest of time, can you answer there and we will move to the next speaker, please? Great. Thank you.